Assalamu alaikum, my dear students. I'm Professor Dr. Zafar Hussain, and I'm talking on the topic of spermatogenesis and oogenesis. And I beg your concentration, please. The topic is very much essential. Highlights structure of a mature spermatozoon, spermatogenesis, oogenesis, ovulation, abnormalities in formation of gametes. Highlights spermatozoon, head, neck, middle and principal piece. Spermiogenesis, spermatid spermatozoon, ovarian follicle. Theca interna secretes estrogen. Ovulation, process of shedding of the ovum. Corpus luteum is formed by enlargement and transformation of follicular cells after shedding of the ovum, secrete progesterone. These are the highlights. Structure of a mature spermatozoon. You are looking here, it's a figure. Spermatozoon consists of a head, neck, middle piece, principal piece, tail. An axial filament passes through the middle piece and extends to the tail, measures about 60 micrometer in the length. You are looking here the structure as well. Well, this is separately shown. The head of the sperm is a spermatozoon, piriform in shape, measures 4 micrometer in length, derived from the nucleus, contains 23 highly condensed chromosomes covered by a cap-like structure called the acrosome. Acrosomic cap, Gallia capitis. The acrosome contains enzymes that help in the penetration of the spermatozoon into ovum during fertilization. Acrosome is derived from the Golgi complex. Well, the neck is narrow, contains a funnel-shaped basal body and a spherical centriole. You're looking here. The basal body is also called connecting piece because it helps to establish an intimate connection between the head and the remainder of the spermatozoon. Axial filament. You're looking the figure over here. The acrosome, nucleus, centriole, coil of mitochondria, middle piece, axial filament, tail sheath and piece. Axial filament, Axial filament begins just behind the centriole, passes through the middle piece and most of the tail, passes through the annulus ring-like structure at the point where the middle piece joins the tail. In the middle piece, it is surrounded by a spiral sheath made up of mitochondria. Is composed of several fibrils. There is a pair of central fibrils surrounded by 9 pairs, doublets arranged in circle around the central pair. This is the pattern of any flagellum. The whole system of fibril is kept in motion by a series of the coverings. Spermatogenesis, this slide should be seen with a keen interest, takes place only during reproductive period, which begins at the age of puberty, 12 to 16 years, and contains even though through old age. मेल में menopause टाइप चीज कोई नहीं होती, दो एंड्रोपास की टर्म आई है, but it is not still proved. The spermatozoa are formed in the walls of the seminiferous tubules of the testes. Testes is called the coiled structure with the seminiferous tubules. The various cell stages as follow. The spermatogonia type A or germ cells, 44 autosomes plus X and plus Y divide mitotically to give rise to more spermatogonia type A and type B. Spermatogonia type B, 44 autosomes plus X plus Y enlarge or undergo mitosis to form spermatocytes. Primary spermatocyte having 44 autosomes plus X plus Y divide to form secondary spermatocyte. This is the first meiotic division. Each secondary spermatocyte has 22 autosome plus X or 22 autosome plus Y. Chromosome, it divides to form two spermatids. This is the second meiotic division. Each spermatid 22 plus X or 22 plus Y gradually change its shape to become spermatozoon. This is called spermiogenesis. Spermiogenesis or spermatogenesis, the process by which a spermatid becomes a spermatozoon. The spermatid is more or less circular cell containing a nucleus, Golgi apparatus, centriole, and mitochondria. All these components take part in forming the spermatozoon. The nucleus forms head. The Golgi apparatus is transformed into acrosomic cap. The centriole divides into two parts. The axial filament appears to grow out of them. The process of spermatogenesis, including spermatogenesis, spermiogenesis requires about 2 months for its completion. So looking here, 16 days, spermatogonium, it takes mitosis, primary spermatocytes. After 16 days, first division. So looking here, the secondary spermatocyte, second division. After 16 days, that forms the spermatid. Maturation and capacitation of spermatozoa. When first seen in seminiferous tubules, spermatozoa are immature, non-motile. In seminiferous tubules, spermatozoa are immature, non-motile, incapable of fertilizing an ovum. Stored in the epididymis, undergo maturation. In epididymis, they undergo maturation. Most of the cytoplasm is shed, but the cell membrane persists as a covering for the spermatozoon. After maturation, they acquire some motility, become fully motile only after ejaculation, when they get mixed with secretions of the prostate gland and the seminal vesicle. ये ejaculation की तारीफ है कि after maturation they acquire some motility, become fully motile only after ejaculation, जब वो epididymis को छोड़ते हैं, when they get mixed with secretion of the prostate and the seminal vesicles. Maturation or capacitation of spermatozoon acquire the ability to fertilize an ovum only after they have been in the female genital tract for some time. This final step is called capacitation, occurs in the uterus or uterine tube under the influence of the substances secreted by the female genital tract. ये बात याद रखिएगा कि capacitation of the sperm occurs in the uterus or uterine tube under the influence of substances secreted by female genital tract. The glycoprotein coat and seminal proteins lying over the surface of the spermatozoon are altered in the capacitation. Maturation and capacitation of the spermatozoa, acrosome reaction. When spermatozoon becomes in contact with the zona pellucida, zona pellucida is a tough layer around the oocytes, secondary oocyte. Changes takes place in the membrane of the acrosome and enable the release of the lysosomal enzymes. This is the acrosome reaction. फिर बोलता हूँ, when spermatozoon becomes in contact with the zona pellucida, बहुत सारे sperm जो हैं वो attach करते हैं, but only one is crowned. Changes takes place in the membrane of the acrosome and enable the release of lysosomal enzymes. वहाँ से लाइसोसोमल एंजाइम निकलते हैं. Zona reaction, some enzymes help in digesting the zona pellucida and in penetration of the spermatozoon through it. You are looking here, acrosome, intact sperm, zona pellucida binding, acrosome reaction, zona pellucida penetration, lemma fusion, intravitelline processing. देखें कितने स्पर्म आए हैं, and only one is going through the zona pellucida by the acrosome reaction and go into the nucleus of the ovum. Differences between spermatogenesis and spermiogenesis, ये गौर से देखिएगा. Spermatogenesis is the complete process of formation of a spermatozoon from a spermatogonium. Spermatogenesis is the complete process of formation of spermatozoon from a spermatogonium. It includes first and second meiotic division plus spermiogenesis. But spermiogenesis is the process of transformation of a rounded spermatid into a spermatozoon with tail and head. ये sperm की बात खत्म हुई है. अब I'm going to start the oogenesis, the formation of the oocytes, the female gametes. Oogenesis. Ovary is the female gonad. Testis male gonad है. Ovary is the female gonad. It has an outer part called cortex. It has inner part called the medulla. The cortex contains many oogonia. All oogonia are produced at a very early stage, possibly before birth. जब शिक मातर में बच्ची होती है तो उसमें oogonia प्रोड्यूस होते हैं in terms of a lakhs. जब पैदा होती है बच्ची तो 1 लाख के करीब होते हैं और फिर जो एक्ट्रेजिया होके 400 या 600 रह जाते हैं and do not multiply thereafter. Oogenesis is similar to spermatogenesis, however, there are important differences as well. See please on this slide. Difference between spermatogenesis and oogenesis. One primary spermatocyte give rise to the 4 spermatozoa. ये याद रखने वाली बात है. One primary spermatocyte give rise to 4 spermatozoa. One primary oocyte forms only one ovum. लेकिन ये बात गौर से कि एक प्राइमरी स्पर्मेटोसाइट 4 स्पर्मेटोजोआ दे रहा है, जबकि वन प्राइमरी oocyte form only one ovum. When primary spermatocyte divides, its cytoplasm is equally distributed between the 2 secondary spermatocyte formed. When the primary oocyte divides, almost all its cytoplasm go to the daughter cells, which forms the secondary oocytes. The other daughter cell, first polar body, receives half the chromosome of the primary oocyte, but almost no cytoplasm. पोलर बॉडी में साइटोप्लाज्म नहीं जाता, सिर्फ क्रोमोसोम्स जाते हैं, न्यूक्लियर मटेरियल जाता है. द फर्स्ट पोलर बॉडी इज देयरफॉर फॉर्म्ड merely to get rid of unwanted chromosomes. Further details. Only 400 ova are discharged during the entire reproductive life of a female. 40,000 ओवा में से 400 जो हैं वो डिस्चार्ज होते हैं in the entire reproductive life of a female. 5 to 30 primary oocytes start maturing each menstrual cycle, but only one of them reaches the maturity and is ovulated. ये मैंने जो फॉर्मूला बताया था आपको कि स्पर्मेटेड स्पर्म भी हैं, कम से कम हंड्रेड्स, इन टर्म्स ऑफ हंड्रेड्स जो हैं, बल्कि इन टर्म्स ऑफ थाउजेंड्स, वो दे ऑल hover around the ovum, but one is crowned. In late fetal period, primary oogonia enlarge to form primary oocytes. At the time of birth, all primary oocytes are in the prophase of the first meiotic division. Their number is about 40,000. यानी जब बच्ची पैदा होती है तो बच्ची के अंदर जो oogonia होते हैं वो अरेस्ट हो जाते हैं फर्स्ट meiotic division में. The primary oocyte remains in prophase and do not complete their first meiotic division until they begin to mature and are ready to ovulate. Further details: the reproductive period of a female is between 15 से 50 years of age. ये है reproductive period. आपसे सवाल हो जाए कि what is the length of the reproductive period? That means कि you can reproduce the replicas 15 साल से लेकर 50 साल तक. This is the age where the lady can have a fertilization and she can give birth. With each menstrual cycle, a few primary oocytes, about 5 to 30, begin to maturate and complete first meiotic division shortly before ovulation. The first meiotic division of the primary oocyte produces two unequal daughter cells. Each cell has the haploid number of the chromosome 23. The large cell is called the secondary oocytes and the smaller one is called the first polar body. The secondary oocyte immediately enters the second meiotic division. Ovulation takes place while the oocyte in metaphase. The secondary oocyte remains arrested till fertilization occurs. The second meiotic division is completed only if the fertilization takes place. याद रखिएगा कि ये जो secondary oocyte है इसकी second meiotic division आफ्टर फर्टिलाइजेशन होगी जिसमें एक पोलर बॉडी बनेगी और अगर ऐसा नहीं होगा तो इट विल बी गोइंग थ्रू द डेब्रिस ऑफ द मेंस्ट्रुएशन. दिस डिवीजन रिजल्ट्स इन टू अनइक्वल डॉटर सेल्स. द स्मॉलेस्ट cell is called the second polar body and the first polar body may also divide during the second meiotic division. If fertilization does not occur, the secondary oocyte fails to complete the second meiotic division and degenerate in about 24 hours after ovulation. Formation of the ovarian follicles. In the stroma of the ovaries, ova develop from oogonia. Then oogonia are surrounded by other cells that form the stroma. These stromal cells form ovarian or Graafian follicles that surround ova and protect them. The stages in the formation of follicle are as follows. Follicular cells. Some cells of the stroma become flattened and surround an oocytes. These flattened cells form the ovarian follicle and therefore called follicular cells. Primordial cells and flattened follicular cells become columnar. Zona pellucida, a homogeneous membrane appears between follicular cells and the oocytes. Membrane granulosa. The follicular cells proliferate to form several layers. These constitute the membrana granulosa and the cells called granulosa cells. You are looking here. Antrum, mature follicle, secondary follicle, primary follicle. These are the developmental changes. Again, you are looking here. Mature vesicular follicle, theca interna, theca externa, antrum, ये cavity, fluid filled cavity. Secondary oocytes, उसके इर्द गिर्द जो सेल हैं, they form the corona radiata. उनको granulosa cells भी कहते हैं. आगे cumulus ovoricus, जहाँ पे वो अटैच करता है एंड पर. Zona pellucida is also there. You can see also here oocyte, follicular cells, primordial germ cells. यहाँ पे फिर देखें आप कि antrum is shown और एक साइड पर the cumulus ovoricus जिसके अंदर मौजूद है secondary oocytes with granular cells. The stages in the formation of follicle are as follows. Antrum, a cavity appears within the membrana granulosa. With its appearance, a follicle is formed. जो ही antrum बनता है, एक cavity बनती है, fluid के साथ हम structure कहते हैं follicle is formed. Cumulus ovoricus or cumulus ovaricus. The cavity of the follicle rapidly increases in size. The wall of the follicle becomes thin. The oocyte lies eccentrically in the follicle. एक तरफ को lie करता है, surrounded by some granulosa cells that are given the name cumulus ovoricus or cumulus ovaricus. The cells that attach to the walls of the follicle are given the name discus proligerus. Discus proligerus. The stages in the formation of follicle are as follow. Theca interna. As the follicle expands, the stromal cells surrounding the membrana granulosa become condensed to form a covering called theca interna. The cells of the theca interna later secrete a hormone called estrogen and they are called the thecal gland. Theca interna cells जो हैं वो estrogen पैदा करते हैं. Fully formed ovarian follicle. Outside the theca interna, some fibrous tissue becomes condensed to form another covering of the follicle called theca externa. This is a connective tissue layer. Interdependent of oocytes and the follicular cells which surround the oocytes. The follicular cells secrete meiotic inhibitory factors. These factors prevent primary oocytes from maturing beyond the prophase of the one meiotic division. This effect may last for many years. The follicular cells are also responsible for growth, metabolism, and maturation of the oocytes. Conversely, the oocytes are responsible for proliferation and differentiation of the follicular cells. oocytes जो हैं वो proliferation भी करते हैं और differentiation भी करते हैं. किसकी? follicular cells की जिन्होंने उसके इर्द गिर्द हाला बनाया हुआ है. Now, factors produced in the oocyte help in the formation and maturation of the Graafian follicles. Ovulation. The shedding of the ovum from the ovary is called ovulation under the effect of LH. The ovarian follicle is at first very small compared to the thickness of the cortex of the ovary. As it enlarges, it reaches the surface and forms a bulging. Ultimately, the follicle ruptures and the ovum is shed from the ovary. Just before the ovulation, the follicle may have a diameter of 15 millimeter, 15 millimeter. The stroma and theca become very thin, and a vascular area, stigma, appears over the most convex part of the follicle. At the same time, the cells of the cumulus ovaricus become loosened by accumulation of intracellular fluid between them. Factors that lead to ovulation: high LH, luteinizing concentration, increased activity of collagenase enzyme, digest the collagen fibers surrounding the follicle, increased prostaglandin concentration results in smooth muscle contraction, increased pressure of fluid in the follicular cavity, and enzymatic digestion. These are the factors that lead to the ovulation at the 14th day of the ovarian cycle. High LH luteinizing hormone concentration, increased activity of the collagenase enzymes, digest the collagen fibers surrounding the follicle, increased prostaglandins concentration, results in smooth muscle concentration, increased pressure of fluid in the follicular cavity, enzymatic digestion, the main factor responsible for ovulation. Structure of the ovum, जो कि shed होगा. Shed ovum is not fully mature. It is surrounded by zona pellucida. Some cells of zona radiata can be seen sticking to the outside of the zona pellucida. It is in fact a secondary oocyte that is undergoing division to shed off the second polar body. No nucleus, nuclear membrane dissolved for the second meiotic division. A spindle is present. You're looking here. 3 months intrauterine oogonium, ये शिक्म मादर में. Primary oogonium, शिक्म मादर में. All primary oocytes at the time of a birth of the baby. फिर first meiotic prophase उस स्टेज में ये अरेस्ट हो जाता है 10 to 40 years तक. फिर meiosis begins again at ovulation. First polar body खारिज हो गई and second meiotic division में second polar body. लेकिन second polar body उस वक्त बनेगी जब fertilization process होगा. Structure of the ovum. A distinct perivitelline space is between cell membrane, vitelline membrane and the zona pellucida. Zona pellucida के दरमियान और ovum के दरमियान एक स्पेस ऐसे कहते हैं पेरीवाइटिलाइन स्पेस. It is known as a peривitelline space. Is between the cell membrane and the zona pellucida. The first polar body lies in this space, peривitelline space. The ovum is very large and measures 10 micrometer. Most other cell body measures less than the 10 micrometer. The ovum is very large cell and measures 100 micrometer. 1 micrometer is equal to 1 millimeter is equal to 1000 micrometer and 1 meter is equal to 1000 millimeter. You are looking here, this is a very good step how the things takes place in the oocytes, granulosa cells, and then antrum, Graafian follicle, then theca cells, and then there's a ovulation at the end. Ovulation takes place. Fate of the ovum. Shed ovum is carried into the uterine tube. The ovary is closely embraced by the fimbriated end of the uterine tube. The ovum is carried into the tube partly by the follicular fluid discharged from the follicle and partly by the activity of the ciliated cells lining the tube. The ovum slowly travels through the tube toward the uterus, taking 3 to 4 days. If the ovum is fertilized, it travels to the uterus and gets implanted in its wall. If the ovum secondary oocyte is not fertilized, it dies in 12 to 24 hours. It passes through the uterus to the vagina and discharged. You're looking here. Well, this is a very beautiful ovulation, fertilization day 1, proliferation, blastomere day 4, differentiation, blastocyst day 5, implantation day 6. You are looking here how this travels. Ovulation के बाद ovum कैसे जा रहा है uterine tube में by the fimbria, and how the fertilization takes place in the uterine tube, and then the zygote travels and get implanted. Corpus luteum, few words, yellow body. It is known as a yellow body. It's an important structure. Main function progesterone secretion, but also secretes some estrogen derived from the ovarian follicle as follows. The follicle ruptures, collapsed and folded. The follicular cells rapidly enlarge and increase in size, acquiring polyhedral shape. The cytoplasm become, देखें जब शेड हो गया ओवम तो जो पीछे स्ट्रक्चर रह गया उसको हम कहते हैं कॉर्पस ल्यूटियम, यानी खाली लिफाफा, खाली बर्तन. दैट इज नोन एज अ कॉर्पस ल्यूटियम. ल्यूटियम क्यों कहा? इसमें पीला जो मटेरियल है वो जमा हो जाता है. येलो. Follicular cells rapidly enlarge and increase in size, acquiring polyhedral shape. The cytoplasm become filled with yellow pigment called lutein, and the follicular cells called the luteal cells. Some of theca interna cells contribute to the corpus luteum. Theca externa का रोल नहीं है, theca interna. Blood vessels from the theca interna invade the corpus luteum and provides it with rich supply of blood. The ovarian follicle itself has no blood vessels, but the surrounding theca is full of them. Subsequent fate of corpus luteum. Next slide. Subsequent fate of corpus luteum. Subsequent fate of corpus luteum depends on whether the ovum is fertilized or not. If the ovum is not fertilized, the corpus luteum persists for 14 days. During this period, it secretes progesterone. It remains small and is called corpus luteum of menstruation. At the end of functional life, it degenerates and forms a white fibrous mass called the corpus albicans, white body. अगर fertilization नहीं होती तो ये एक white body corpus albicans बन के ख़त्म हो जाता है. It remains small and is called corpus luteum of menstruation अगर fertilization नहीं होती. Subsequent fate of corpus luteum. If the ovum is fertilized, In pregnancy results, the corpus luteum persists for 3 to 4 months. The larger than corpus luteum of the menstruation and called the corpus luteum of pregnancy. अब ये corpus luteum of pregnancy को कहते हैं, ये large होता है corpus luteum of menstruation से. The corpus luteum of pregnancy may occupy 1/3 to half the total volume of the ovary. The progesterone secreted by it is essential for the maintenance of the pregnancy in the first few months. After the 4th month, the corpus luteum is no longer needed as the placenta begins to secrete progesterone. Human chorionic gonadotropin secreted by the trophoblast cells of the developing embryo prevent degeneration of the corpus luteum of pregnancy. Here, गौर रहे इस बात पर. Human chorionic gonadotropin HCG secreted by the trophoblast cells of the developing embryo prevent degeneration of this corpus luteum of pregnancy. The series of changes that begin with the formation of an ovarian follicle and end with the degeneration of the corpus luteum is called the ovarian cycle. Fate of the ovarian follicles. In each cycle, only one follicle reaches maturity. ये बात याद रहे. In each cycle, one follicle reaches maturity and becomes a corpus luteum. At the same time, several other follicles also begin to develop, but do not reach maturity. These follicles do not persist into the next ovarian cycle, but undergo degeneration, which is known as atresia. The ovum and granular cells of each follicle disappear. Thicker internal cells proliferate to form the interstitial glands corpus atretica. These glands are believed to secrete estrogen. After a period of activity, each gland becomes a mass of scar tissue indistinguishable from the corpus albicans formed from the corpus luteum. Differences between male and female gamete, इसमें देख लीजिए, मोटिलिटी है, साइज एंड शेप है, साइटोप्लाज्म है, क्रोमोसोमल टाइप है. You can look upon here. Testes, spermatogonia, spermatocyte, meiosis, then spermatogenesis, sperm, and you know in ovary, oogonium, oocytes, then oogenesis, वहाँ पे एग बन गया, यहाँ पे स्पर्म बन गया. Reproductive